Hello. Yeah. Good morning, everyone. I am Dipansh Rajput, one of the GC of Mimamsa 2024. I welcome every one of you here for the deep thought of biology. As we know, these are the 4 teams which are competing in mains. So this is IISc Bangalore sitting at the rightmost position. Then after that, there are IIT Bombay, then IIT Delhi, and then at last IIT Kanpur. So now I would like to introduce our judges. Our judges for biology deep thought are Professor Emeritus Vinita Bal and Dr. Suhita Nadkarni. Professor Vinita Bal is a scientist at National Institute of Immunology. Okay. Yeah, he is a faculty of IISER Pune only. Her work involves analysis role of T lymphocytes in pathopsychological conditions. Dr. Suhita Nadkarni from IISER Pune is a Wellcome Trust DBT fellow. She works on biophysical model of synaptic plasticity in health and disease. Please have a great round of applause for both of them. I want to thank both of these. These 2 people are there for during the framing of questions. I want to thank on behalf of our question making team. Thank you for being there, and thank you for being here as a judge. Now, for the audience and the participants and the judges, I will briefly go through the rules and the guidelines. So this deep thought will be of 2 hours per subject. The maximum marks, there will be one directed team which is directed towards the question. They will choose one question out of 4. That directed team, if they answer correctly, they can get maximally 20 marks. Then after 15 minutes of time, this question will be passed on to the another team, and their time will be 2 minutes to present. That team, if answer correctly, they can maximally get 10 marks. After this will be passed on to the next team, and if the next team answer correctly, they can get 5 marks. Same with the another team, they will get 1 minute to present, and maximum marks will be 5 marks. There will be, for time, there will be initially 3 plus 1 minute time for presentation, 3 minutes time for the podium coordinator will present the question. After that, for 1 minute, there will be clarification time for directed team only. If directed team have any, they need any clarification, they can just ask the QMCs. QMCs will resolve their doubt regarding the question. After that, the 15-minute timer will start. There won't be any a single second gap in between the passing of the questions. The immediately question will be passed to the next team. Please ensure participants should ensure few things. They should not repeat the previous solution. Whatever presented by the previous team, that won't be marked or that won't be graded. So another thing, and any decision made by our judges and the question making team will be final decision. Now I will ask our podium coordinators to come and take over for the biology deep thought. Please.
Good morning everyone and welcome to Biology Deep Thought. I am Urvija and this is Shritama, and we will be our podium coordinators for the day. We would like to welcome our judges Ms. Vinita Bal and Ms. Suhita Nadkarni, and our QMCs Ameya and Suhani. We would request the judges to pick a chit to decide the order of the event. Okay, so it will be IISc Bangalore first, followed by IIT Bombay, IIT Delhi, and then IIT Kanpur. IISc Bangalore, please pick a question of your choice out of these 4 questions. Okay, you're going with deceased and diseased. IISc Bangalore, your question is, 2 genes, A and X, were studied in a family of aliens. Sex determination works by the same mechanism as in humans. One of them is present in the tryptyl, a haploid organelle 1 per cell, and the other is in an autosomal gene in the nucleus, diploid 1 per cell. Tryptyl is provided during fertilization only by the ovum. Gene A has two forms, A, which is never methylated, and B, which gets methylated or silenced. Unmethylated B is the same as A, which produces a transcription factor A. A decreases transcription of the gene X by 50%. If a pregnant female is starved during the early part of her pregnancy, B in somatic cells of the embryos does not get methylated, but in the gametes of the embryo, B still gets methylated like it should. The pregnant female herself is not affected by starvation. The wild-type allele of gene X has no effect on anything else. Small x is the mutated allele of gene X such that 100% expression of one copy of small x leads to the individual dying due to a metabolic disease after 35 years. More expression of small x than in the above case leads to severe metabolic disease and the individual dying immediately after birth. Such individuals do not exist in this pedigree. 50% expression of one copy of X makes the individual have the metabolic disease, but the individual does not die. Turning is a rare phenomenon where a cell, heterozygous for a gene, becomes homozygous for it, but it does not affect the gametes. There is only one individual who is a descendant of generation 1, but not a part of generation 1, with turning in the cells affected by this metabolic disorder. Only one of the female in generation 1 was starved during the early part of her pregnancy. Assume all children of a particular set of parents come from the same pregnancy and all are born to the parents before the age of 35. Kindly answer all these questions. Give possible genotypes of all individuals. Please note that you need not make the pedigree again while presenting your answers. You can use the numbers given to give the genotypes of all individuals. Which gene is in the second question is, which gene is in the nucleus and which one is in tryptyl? Third is, which female was starved and which individual shows turning? IISER Bangalore, do you want some clarifications? Okay, your time starts now.
Pedigree was because see, it's an easy question and it's a very hard question to cover. And there is a lot. But good. That is this handicap, which is verbal expression and intelligent emotion is very continuous manner. Yes. उनकी जो हंसी में ये चूज़ा क्योंकि हंसी कैसी है मैंने भिगा दिया चॉक्स
IISc Bangalore, you have 7 minutes remaining.
Don't let them throw it on the laptop. They did that.
Throw it on the laptop. What the fuck? What did you do that? Like something already. But it's hard by the way.
So don't trust your mind. Suddenly I'm like,
Right now I was already with someone.
last was
IISc Bangalore, you have 5 minutes remaining.
करते रहते हैं बार बार बोलते हैं पर ध्यान रखते हैं जल्दी सीधा बायो का है फिजिक्स का डिस्कसिंग मैथ्स का discussing pedigree research. Let's talk about technical to biology. That is like something. I don't know what are they going to do.
You can just say it, you don't have to write it.
Okay. Yeah, so we think that X is in the autosomal. Right. We think X is an autosomal gene and A is a gene in the triptyl. And so since the triptyl is haploid, you have just one copy of A in the triptyl. And if you have and you have two copies of X in the autosome, and if A is methylated, then you don't make the transcription factor, and you will have the disease. And if A is not methylated, then you will make the transcription factor, but you won't die if you have the small x. Since in the generation 2, in the like 2, 1, 2, 2 and 2, 3 and 2, 4, they are the descendants of the 1, 1 and 1, 2. None of them have the disease. We think female and generation 3 has generation 3, 1 has disease and 3, 2 has a disease and 3, 4 has a disease. We think female 2 was starved. And
Last 40 seconds.
okay. And we think generation 9 probably, generation 2, 9 probably showed the turning because it turned from heterozygous to homozygous, and it started having disease and it died. Yeah.
IISER Bangalore, your time is up.
इसमें जो लिखा है वो लिखो।
Yes. So for the possibilities of genotypes of all individuals, we are writing them down over here. Which gene is in the nucleus capital X and which one is in the triptyl A? Because there is one condition where there is less than 50% expression of one copy of X, which is only possible if capital X is in the nucleus. As per the diagram that they are making right now, the female which was starved, according to us, it should be 4, and the individual which shows turning should be 2.
And the reason for the individual 2 showing turning is because the children of 2, that is 1, gets dies after 35 years. So one of the like this mutated allele should be in 1, and as a result, the 2 would have turned homozygous from its heterozygous condition. Like I'm talking about the individual 2 of generation 2. Yeah. Sorry? Individual 4 of generation 1.
Last 20 seconds. IIT Bombay, your time is up.
For the second question, we'll say that the gene in the nucleus is capital X and the gene in the triptyl is capital A. Which female was starved should be female 4 because it's in generation 1 because its partner is shows 100% expression of X, but none of its children are diseased. which individual shows turning, we'll say individual 5 in generation 2, because again, its parent 3 shows 100% expression of X, but none of its descendants is diseased. So it should have turned with respect to gene X.
IIT Delhi, your time is up.
So first off, the gene that is present in the nucleus should be capital X, because there are cases where there is less than 50% expression of copy of X. It is only possible when it is, say, heterozygous in X. So now, in the first generation, for individual 1, the genotype should be like small x, capital X, and A. So there is less than 50% expression. Then the female which was starved should be female 4, because its partner is completely deceased, and some of the descendants are also deceased, so which means that some of its children should have gotten the B form, which is producing somatic cells due to starving. So,
IIT Kanpur, your time is up. Judges, do you have any questions for our teams?
Okay. Is there an example of turning in the real life that you are aware of? Anybody can answer and you will get marks. Did you get the question? This was a hypothetical exercise, right? But we have genome, we have everything. So in that sense, do you know of an example where turning happens?
So I think I have heard something like some species of frogs turn their sex depending on the environment and conditions. So that may be a example of turning in real life.
Elaborate a little bit on what you mean by environmental conditions. So is this a genetic change? Is this phenotypic change?
It may be phenotypic change. I'm not sure on that.
Okay. Similarly, what was it, the word? Truptial be representing in real life?
It can be representing mitochondria in real life.
Fair enough. All right. We'll change it.
Ma'am,
Yeah.
one thing, can the turning in the fishes, when the clownfish, the female is not there, the male turns into the female, it can be
I
considered?
think it's a similar situation. Okay.
I now request someone from the question making team to present the solution.
Hello. Okay, so according to our answer, the A gene is in the nucleus and the X gene is in the truptial. The reason for that is from the pedigree chart and the starvation. So starvation is basically you have the gene, the allele A or the version of gene A and B. This one is methylated under normal conditions, but when you are starving a pregnant female in the early part of her pregnancy, the embryo does, the somatic cells of the embryo remove this methylation and then it acts like A. However, when the gametes are produced in that embryo, they still have the methylation. So the reason for saying this is in the nucleus is because so the most reasonable case to consider to find out most of the information would be the one where you have all three phenotypes in the same set of children. So when you do that, So you would have BB over here. So according to this model, AA plus small x would give you the normal phenotype where nothing dies. AB plus X would give you diseased phenotype, which is this one. And BB plus X, where B is methylated and both these B's are methylated, it would give you dead after 35 years. So this would be BB plus X, AB plus X, and AA plus X. And accordingly, so here is why we think the generation 1, 2 female is starved. If she is starved, she has to have X because it is maternally inherited. Because we're saying this is in the Truptial. So she has X, so all her children have X. So since we know that these have to have X, she must have an X and she must have an X. Now he has AB, and these children have BB, AB, and AA. So both of these have to have AB. And so now this AB, so if it was AB plus X normally, she should be diseased. However, she is not diseased. So that means the B methylation is removed in this. So if you starve a female from the first generation, the children in the second generation will be affected. So the B methylation in the second generation gets removed, but the third generation still has methylation. So her methylation is removed, so she has a normal phenotype, while her children have methylated Bs over here. And so you can apply that to the entire pedigree. And the reason the other way won't work is because you would have strange combinations of X, like, so if you had X, capital X, small x, B, BM, then X would be expressed. This is dead. However, then this would have to be X plus A, because this would decrease the expression of the small x by 50%. So this has to be the disease phenotype, and this has to be the dead phenotype. But now the Truptial has B and A in the same set of children, which is not possible. Now the turning is applied to generation 3, 10, and so that is because so you have BB over here and BB over here. Now BB plus X and BB plus X. So this person has X. Now the only way this can have 2 copies of B is if this person also has a B. From this, the 1-4 female, she will have capital X. So this person will have plus capital X. That's why he, even despite having a B, he does not show any phenotype. It's not small x. And so this person has to have an A, at least one. Now, we are saying that this person, which is 310, is turning. So he turns to AA plus X. She turns to AA plus X. Now, the reason we're saying that, this has to have a B. So the only options are BB plus X or AA plus X. If it was BB plus X, this would also be BB, and this would die. So the reason this individual has to be AB plus X, AB plus capital X. So then you get AB over here and BB over here. And then this individual shows turning into this. And the rest, if you apply this, this model to the rest of it, you will figure out the genotypes.
Hello. IIT Bombay, you're up next. You may choose between the questions 2, 3, and 4. Your question is,
In a species of rats with XY sex determination, two kinds of males exist in the population. Males can be monogamous, that is, mate with only one female the entire mating season and stay beside her always to deter other males, that is, guard them, or be polygamous, that is, leave a female after mating with her and move around to find other available females. Whether a male indulges in monogamy or polygamy is genetically determined by a wilding trait. Females can mate with any male who approaches her depending on the state she is in when a male arrives, which may be available for mating. She may be pregnant, that is, if the female gets pregnant at any point, she is not available for mating for the rest of the mating season. She may be in refractory period, which is a period shorter than the mating season after a mating in which the female rejects all males till she finds out whether she is pregnant or not. One mating season can accommodate multiple refractory periods. The mating season lasts for the entirety of spring. There are 4 isolated regions. In region 1 and 3, the male takes a long time to find a female to mate with due to low population density. In region 1, there is a long spring and region 3 has a short spring. Similarly, in region 2 and 4, the male takes a short time to find a female to mate with due to high population density. Region 2 has a long spring, whereas region 4 has a short spring. 4 identical populations with equal numbers of males and females are placed in each region. Assume polygamous and monogamous males are equal in the population initially. Assume fitness of an individual to be the number of successful matings, that is, matings which result in impregnation. Assume the rats live for only 1 year and there is only 1 mating season per year. And assume no parental care by any kind of male. And assume no abortions. Let del F be the average fitness of polygamous males minus the average fitness of monogamous males. Question 1. In which region would del F be highest and where would it be lowest on the average after 1 mating season? Explain. Now take a new population of the kangaroo rats present in a particular area with a fixed mating season, enough to accommodate multiple refractory periods. Question 2. How does del F change over a mating season? That is, what does the graph for del F versus time look like for 1 mating season? How does the change in del F over a mating season vary for populations with different sex ratios? Explain. Another population has a sex ratio female biased enough to ensure that most males find at least 1 mate. The time taken by males to find females is short enough to ensure females are not mate limited, except when all males are monogamous. There is a fixed mating season which can accommodate multiple refractory periods. Question 3. How does expected del F for any 1 mating season vary with the frequency of polygamous males in a population? Give the shape of the graph for del F versus frequency of polygamous males in a population and explain. IIT Bombay, do you want some clarifications? Your time starts now. IIT Bombay, you have 7 minutes left.
Delta F will be highest in region 2 and the lowest in region 3 on average after one mating season. This is because in region 2, the polygamous males will take the least time to find a female to mate with, and there is high population density and a long spring, so they'll be able to mate with more females on an average. While in region 3, a polygamous male will take much more time because of the low population density and it's a short spring.
3.5 minutes are remaining.
So for the graphs of delta F over time, for the region 1, it will, first of all, it will be 0 because it takes a lot of time to find the females. But eventually it will sort of increase and attain a saturation point where the number of females is, basically they're set into the refractive period. For the region 3, it will only increase by a little bit and there will not be much time to find the females. So the graph will look, it will just be a straight line and it will not have any interesting characteristics. For the region 2, it will increase because there is a large population density, but when the refractive period sets in, it will sort of dip down. But depending on whether the female is pregnant or not, it will increase and sort of oscillate and attain a saturation point. For the region 3, again it will look like region 1. For the region 3, we don't have much time because the spring is short, so it will just increase and attain a saturation point early, and there will be no dips because the refractive period is quite short here.
One 1.5 minutes remaining.
The final graph for the third question will look something like this. The graph will be the most negative when the frequency of polygamous males is 0, because there are literally no polygamous males. And the frequency of the polygamous males will at max, when the frequency of polygamous males is 1, the average fitness will be the highest, and the graph will look like an exponentially increasing curve, because as in when the number of polygamous males are increasing, since there is a very female biased sex ratio, all of them will always find a female to mate with, and as there are more polygamous males, they will find more females to mate with, and so the impregnations will be higher. It will be exactly 0 at a point at which the frequency is greater than half. It won't occur exactly at half.
Twenty seconds remaining. IT Bombay, your time is up.
So for the second part, we know that when the sex ratios are roughly equal, then at the start, both monogamous and polygamous males will find a roughly equal number of females, and the ratio will be 1 at the beginning. But over time as well, we'll say that because we know that finally we'll also go to ratio 1 when the sex ratio is also 1. We believe that the number of pregnancies that will happen will be similar as well. So the graph will be constant. When the sex ratio is in fact more females than males, then we'll start off 1 again in a similar case. But we will increase because there are a number of polygamous males remaining will be much more, and then the remaining females will get divided for the polygamous males more. Similarly, when the sex ratio is bad for males, the males are much more, then we'll start off with 1 again and decrease, because anytime a monogamous male files the female, then he's sure to impregnate her. But so after a particular time, when some of the females are remaining, and the monogamous males that find a female will definitely mate with her, but the polygamous males might not impregnate them, and so the graph will be decreasing. And then for part 3.
For the third part, we'll say that initially when there are no polygamous males, the delta F will be very low. Then it will start to increase, it will reach a maximum, and then it will again start to decrease as the frequency of polygamous males reaches 1.
And for part 1, region 3 is lowest and region 2 is highest.
Ideal, your time is up.
Okay, so for the first one, the delta F will be highest in region 2 because it's a long spring and it takes a short time for a female to mate, and it would be smallest for region 3 because a short spring and it takes long time for a female to mate. For region 2, a short time for female to mate. Now, for the second one, for region 1, the graph will look like this because it takes long time for a female to mate, so chances are the polygamous will mate with more females in this region. This is a point where the monogamous rat mates with a female, so it comes down a bit, and then so on. So it goes like this. For region 2, it takes short time for a female to mate, right? So the polygamous would mate first till here, and then after that, it would go on oscillating because both of them would mate with females.
ID Kanpur, your time is up.
Thank you.
So for the third question, you will have the frequency to. So initially, when the frequency is small, that time monogamous females have polygamous males have a lot of advantage. So it will be high, and as the frequency increases, it will decrease. And highest is in region 2. Highest delta F is in region 2 and lowest delta F.
Speak up? It's very hard to, because you're not, the labels are not there, so we're only...
Okay, sorry.
So for the question 2, region 3, it will be approximately 0 because the chances of finding mate are equivalent for both of them. And about the after one refractory period, it will increase a little bit.
IISER Bangalore, time is up. I would request someone from the judges. Do you have some questions for the teams?
Probably a little too easy, but if for the third one, expected delta F for any one mating season. So most of you are trying to tell us for all 4 regions. Is that what you are trying to say? What does the expected delta F for any one mating season? So it was any one mating season. Even for the second one, there was, so second question, now how does delta F change over a mating season? It doesn't necessarily ask you for region 1 versus 2 versus 3 versus 4. That region 1 to 4 was primarily meant only for question 1. So most of your answers got sidelined. Is that what you see? If you reread the question, am I right? Yeah? So if you reread the questions, question 2 and 3 is not focusing on region 1 versus 2 versus 3 versus 4.
There's a general trend across regions.
So
we tried to...
So there was misunderstanding or misinterpretation of what information was given in most of the cases. So all of you have looked for question 2 and question 3 independently for region 1, 2, 3 and 4. That was not intended, at least. Let the people respond.
I would request someone from the question making team to present the solution on the board.
IIT Delhi, do you want to, I didn't understand how the different gender ratios or different ratios would affect your curves. If you read the question, there's a part to it. And I think you mentioned it, but I didn't exactly get it. Question 2. How does it vary with different sex ratios?
Yeah, so what I said was initially both will find roughly similar number of females, both types. So we'll have...
When the initial ratios are different, so when the ratio of male is more than females and less.
Yeah.
So the 3 scenarios where it's equal, less, more.
Yeah, in all 3 cases, since the 2 types of males are equal, they will both find a roughly similar number of females. That's why all the graphs start at 1. That's what I've said. And then over time, when the females are lesser than males, then because once a female mates with a monogamous male, then she will mate with him regardless of whether she gets pregnant or not. Yeah. Okay. Yeah. So the first one, yes. So this is for when the ratios are equal. Yes, and this is when the female ratio is higher than males, and this is when the female ratio is lower. Yeah. Yes. No, I'm saying that delta F will. Oh yeah, sorry. Yeah, that's what I was doing. Yeah. Yeah.
I would request someone from the question making team to present the solution.
Hello. Can you present the question? No, the question. Just the question. Just present the question. Hello. So, as most of you answered correctly in the first region, so basically what happens is, for a polygamous male, the fitness roughly depends on how much time you have left in the breeding season and how much time does it take to find a new mate is inversely proportional to that, roughly. So basically, if you have a high density, you will have to spend very less time in finding a new mate. There's no cost of leaving your mate because you are sure to find a new mate. So in such situations, polygamy is favored. Also, when the breeding season is long, you can have multiple matings. So in that case, there's no point in sticking with your mate. So polygamy is favored. So as you mentioned it correctly, in the region with long breeding season and high population density, polygamy is favored, and vice versa for monogamy. Now let's look at the second part. So it's delta F, by the way, not F. Many of you were plotting F versus time when we are clearly asking for delta F. Basically, how much is the advantage for polygamy at any given time of the mating season, right? So initially, towards the start of the mating season, there are males and females are available, quote unquote available. So, and as time progresses, more and more females exit the mating pool because they are in the refractory period, right? So the sex ratio becomes more and more, the operational sex ratio or the sex ratio of the mating pool becomes more and more male biased as mating season progresses. So essentially, polygamy is favored when you have a fair chance of finding a new mate, which happens when there are available females. So essentially, there is a advantage, not advantage, but like the disadvantage for polygamy relative to monogamy grows as the relative disadvantage grows as the season increases, except for the end of the season when, so think about it in this way, at the very end of the season, there's no point in sticking with your mate because she can't have one more mating. Let's say the refractory period is 8 days and you have found a mate on just 6 days before the end of the mating season. There's no point in sticking with her because you know she can't get pregnant again. So in that case, you have to leave. You have to be polygamous. So this delta F is basically a curve like this, mostly decreasing, but at the very end, there's a terminal increase. Now, how does it change with sex ratio? Except for very highly skewed sex ratio changes, the shape remains the same. It's just that the intercept changes up and down. So when basically, if the population is male biased, polygamy is disadvantageous and it becomes this. When it is female, the curve shifts upwards. This is 0. So only the intercept changes with sex ratio. Right? Now let's look at the third part. Cool. So for this given population, you see, yes, the sex ratio is female biased, which ensures that almost every male gets a mate. So even the monogamous males are getting a mate and polygamous are also getting a mate. So let's look at the overall number of offspring produced in a population. That's something like C. That's constant because that depends on number of females and all females are getting a mate because females are not mate limited. All females are getting a mate. So the number of offspring is constant irrespective of what's the frequency of polygamous males or monogamous males. Now, since monogamous males stick with their mate, let's say each mating leads to X offspring, their fitness is constant basically, X. Let's say P is the proportion of polygamous males, and F is the fitness of polygamous males, average fitness of a polygamous male. So basically, you get this equation. P is proportion of polygamous males, F is fitness of polygamous males. This is the offspring produced by polygamous males. This is the offspring produced by monogamous males, and this is the overall number of offspring. Now, X and C are constants, so if you express it in terms of F, you get basically P in the denominator, so F is this, so you get a hyperbola, which is basically what IISER Bangalore, I think, drew, but they did not call it a hyperbola, but yeah, it's a hyperbola. Yeah. In finite populations, it will saturate at this point because your fitness can't go infinity, but for infinite population, it's a hyperbola. It's not defined at x is equal to 0. It's a singularity there. So I hope the explanation of the solution is clear to everyone. And yeah, don't take any real life advice from this question. Yeah. Thank you.
IIT Delhi, I now request you to choose between second and third question. So the question is as follows. Joe, a newly discovered insect, has box-shaped eyes on either side of his head. Joe is held fixed in a black box with the right and left walls displaying a video of the vertical black and white stripes, perpendicular to the direction of the motion of the video, moving at speed a by t, as shown in figure 2. Width of black stripes is 2a and white stripes is a. Each eye's retina has 6 photoreceptors in 2 rows. Length of field of view of a photoreceptor is equal to the length of each photoreceptor, which is equal to a. One photoreceptor activates the A neuron connected to it only when an increased level of brightness fully fills its field of view. The neural pathway for detection of direction on the left and right side is independent. The figure 4 shows a schematic diagram for neurons between brain and retina for both rows. Each A and B neuron belongs to only one row, while C neurons are connected to B cells of both rows. Observations for neurons between retina and brain upon activation. A2 immediately sends a signal to B with intensity just below the threshold for activation of B, but can cross the threshold due to random fluctuations. A1 and A3, activated and observed separately, send signals which reach the B neuron at a time t after their own activation. At the time when these signals reach B, they only modify the signal from A2, which reaches B at the same instant. B sends a signal immediately to both the C cells along A1 to A3 direction. The 2 C cells can receive a signal from a B at either end. If a C cell on one side of the head is activated, direction of surrounding motion on that side is interpreted as being along the C cell's end 1 to end 2. All neurons can get activated only once the previous signal has been transferred, and all neurons of a given time have the same function in every row. For Joe to be able to detect the direction of apparent surrounding motion, what is the function of the signals from A1 and A3 neurons? How does the signal from a B cell affect each C cell? If the same pathway with all neurons having the same functions could also sense a decrease in brightness, would detection of direction of motion work? Explain. We increase the speed of the right wall from a by t till 4a by t. The left wall remains at a by t. What would happen to Joe's perception of his own motion as the right wall speed increases? Explain at integer multiples of a by t. IIT Delhi, do you want any clarifications? Okay, your time starts now. 6 minutes remaining.
So when the frame is moving towards this eye, first, since it is written that the A neuron gets activated when it first sees a fully bright picture in front of it. So it would get activated when this point, like this is A. So when this reaches here, this A would get activated, and with a time so this is white, this is black. So when this border reaches till here, this neuron would get activated, and it would send a signal to B. After some point, this neuron would get activated, and this would again send a signal to neuron B. So and after that, this neuron would get activated. So since these signals are coming at a time delay t, and they are modifying the responses that B receives, this B can get to know that this was stimulated first and then this was stimulated. And similarly, the B that is here would get to know that this was stimulated after this got stimulated. So in both the directions, it can know in which direction the brightness was moving. So yes, that is for part one. For
Or they have a doubt. They have a doubt. You have 2 minutes remaining.
In the second one, we'll say that Joe cannot detect which direction of motion is working, because assume that first, in one case, the bands of black and white stripes are moving from top to down, and in another case, the bands of black and white stripes are moving from down to up. In bisymmetry, both the cases would look same to Joe, because he can detect both black and white stripes. So we don't think that he could sense the direction of motion.
Okay, so for the last part, as we increase the asymmetry of the speeds in the left and right walls, Joe would feel that it is rotating, but as the speed of the right wall increases, the direction of rotation would also switch, like it happens with humans too. So yeah, that's what we are expecting.
Yeah, so like for the first part, as he said, there will be a time gap of t for transmission. So we would know that when this lights up, after t seconds this will go here, when this would have lighted up. So the function of it, this is to determine like if this lights up first and then this lights up, then we would know the direction is from here to here. And similarly in this case, if this lights up first and this lights up, then also we know that it's from here. So that's why A1 and A3 are changing the signal at B. And for the third case, since the time delay is t, when the speed is 4a by t, what will happen is that some black strips will get skipped, black and white skipped, and it will
Hi, Teddy, your time
perceive
is
same
up.
speed.
So for the first part, what will happen is, so since we have these three neurons like this, so we are going to assume that whenever a bright band is there, we'll assume that the threshold brightness for activation would be like, since the field of view is also A, if say suppose some X amount of this is present in the field of view, we'll assume that the neuron is activated. Now since the width of the bright band is A, so from A1, A2 and A3, only two of them can be activated at the same time. So the function of A1 and A3 is basically such that, so there'll be cases when we have A1 and A2 activated, A2 and A3 activated. So now these things, when they change with respect to time, it can be used to perceive motion, because the signals from A2 and A3 will modify the signal from A2, which will then change the signal which occurs at B. So this is the function of A1 and A3. Now for the third question.
For the third question, basically like for the object, one side would be moving slower than the other side. So the object would perceive it as if the object is rotating. And this is the explanation.
IIT Kanpur, your time is up.
Basically it's a type of lane following robot kind of situation. So for the part one, we don't think it can detect motion, because like the sensors need to be fully covered, as we asked in clarification. But for the part two, where it can sense brightness, it can use the brightness to like control signals from A1 and A3, such that it uses it for sensing forward direction and backward direction by like different kinds of W, black W, and those types of combination. Now for the part 3, since right wall is seen to be moving faster than left wall to the bot, so it happens when we are moving towards left, because if we are rotating, then the motion to the location where we are.
IISER Bangla, your time is up.
When the speed is 4a by t, the perception of motion of the B will be exactly the same, because let us say at t equal to 0, the white stripe reaches the first receptor. Then at t equal to 1 by 4, it will reach the second receptor. T equal to 3 by 4, it reaches the third receptor, and so on. So what would happen is that at t equal to 0, A1 receptor will send a signal that will be received at t equal to time t. And at that exact same time, we will also receive a signal from A2. And therefore, at time t, we will get a signal on B. So it will still have the same speed.
For 3a by t, For 3a by t, no signal will be detected, and for 2a by t, the direction of motion will be opposite. For 4a by t, we'll detect the same direction of motion as initial, and this will repeat in cycles of 3.
IIT Bombay, your time is up. IIT Bombay, your time is up. Judges, do you have any doubts for the teams?
Can you go back and you spoke about how A1 and A2 collaborate and A2 and A3 collaborate, right? Can you, can you take it further? How do they collaborate? I forget what you said. Did you say that the animal can tell the direction or no?
We said it can tell the direction because there is a time lag T between the activation of B due to A1 and A3. So when A3 white light comes in front of A3, after T seconds, it will send a signal to B, at which time A2 will also be activated. So it will know that A2 and A3 are sending activation signals.
So what's the role of A1 as opposed to A3? Do they play exactly the same role?
Yeah, so their role is, their role is similar. Yes.
So exactly the same?
Yeah, they're just for determination of directions.
So then there is symmetry, right? Then how do they, how do they...
They send the opposite direction as in, like if A2 and A1 are activated, it will do the opposite direction. And if A2 and A3 are activated, it will consider the opposite direction basically to the other one.
I now request someone from the question making team to present the solution.
So first, if the fly sees the surroundings all moving backwards, he thinks he's moving forwards. If the directions here are opposite, he thinks he's turning. Now, for the neurons, when A2 sends a signal to B, this is very close to the threshold of activation of B. So we say that A1 modifies the signal by increasing the intensity a little bit so that B is activated, is always activated if these two signals combine. And when A3 sends a signal, it reduces the intensity of the signal from A2 so that B does not activate, and this particular row will just not send a signal. When a bright object is moving in this direction, like the white stripes here, A1 gets a signal at time 0, let's say. The speed of this object is A by t. A2 gets a signal at time t. These signals combine here. The B cells send a signal to C. And when this object reaches here and goes away, this A3 signal comes alone and does nothing. We say that a signal from B will activate a C cell if it reaches N1 and inhibit it if it reaches N2. So this particular row of neurons will detect motion in this direction but not in the other. That's why the other row arranged in the opposite direction will detect the other direction of motion. Now, this particular pathway, if it also sensed a decrease in brightness instead of just increase, at many instances, let's say at time t after this particular situation, the A1 will send a signal for increase in brightness and A3 will send a signal for decrease in brightness. They'll cancel each other out and we will just not detect any motion. That's why this particular thing can only sense either increase or decrease in brightness. Now for the third part, if we when the pattern is moving at speed a by t, after time t, we see this, and then after that we see this. As we've seen before, the fly sees this as motion from left to right. If the pattern is moving at speed 2a by t, the fly sees this first, then this, and then this, which is and the fly would see the same thing if the pattern was moving towards the left with speed a by t. So in that situation, it does see the pattern moving towards the left in the opposite direction with speed a by t, and thus thinks he's rotating. When the pattern is moving at speed 3a by t, it just doesn't detect any motion, and at 4a by t, it goes back to the same situation as a by t.
IIT Kanpur, your question is, a bacterium Corynebacter undergoes undergoes sporulation under adverse conditions as a survival mechanism. The process ends up releasing a copy of the bacterium's DNA into the environment. Some individuals decide to take a risk by not sporulating. Instead, they differentiate into a state called competence, wherein they take up the DNA released by sporulating members of its colony, thus potentially increasing its genetic diversity. Keep in mind that not all cells become competent. Shritama was studying the regulatory mechanism in the bacterium which makes the decision between competence and sporulation. The master regulator of sporulation is the transcription factor R-anakin. Upon exposure to stress, R-anakin is activated by phosphorylation, but at a very slow rate. On reaching a threshold concentration of phosphorylated R-anakin, the bacterium sporulates. They also discover that competence is controlled by a master regulator transcription factor S-anakin, which activates transcription of competence genes once its activity crosses a certain threshold. The sporulation module is inhibited during competence. Confusingly, R-anakin was found to inhibit NEHAES, an inhibitor of S-anakin, but also activate vignin, another inhibitor of S-anakin. An experiment by Shritama revealed that on introduction of stress, there is a certain time window during which a cell can decide whether to sporulate or not. With the above information in mind, explain how this time window forms. What sort of mutations would change the duration of this time window? It was discovered that S-anakin activates its own transcription at all concentrations, thus making it a positive feedback loop. It shows cooperative binding, thus giving us a sigmoid graph of its activity versus concentration. The graph below shows the production rate of S-anakin against its concentration. The graph levels off because of saturation of the transcriptional and translational machinery. S-anakin is actively degraded by the Shrishti complex as shown. The graphs are made at the same scale. All the pathways discussed so far are heavily prone to cellular noise, which are stochastic perturbations. How does the cell become competent? Why doesn't every bacterium in the population become competent? A graphical answer is appreciated. Shrishti is competitively inhibited by paradithin, which can cross membranes and is secreted by the cell at a constant rate at all times. Competent cells do not secrete paradithin. Paradithin has a small lifespan. Explain the role of paradithin and the evolutionary advantage it gives. Do you have some questions? You can see. Okay. Your time starts now. IIT Kanpur, you have 7 minutes left.
Okay. First up for part A, we believe that the time window forms because, as was mentioned, phosphorylation is a very slow process. So as the stress increases and the phosphorylation starts, R anagnin slowly gets converted to R star anagnin, which causes changes in the concentration of both the inhibitors, which is NAHES and Vignin. Now the equilibrium between NAHES and Vignin would actually influence the production of S anagnin. So as is visible from the graph of S-anagnin production versus concentration, so in that graph, if the equilibrium is say attained before the production starts to increase exponentially, say suppose it's attained here, then in that case the production of the sporulation will dominate over competence and the bacteria will do sporulation, whereas if that equilibrium of S-anagnin concentration is say suppose attained here, then the bacteria would undergo competence over say suppose sporulation. So this equilibrium will take time to attain because of phosphorylation being a slow process and for the enzymes to equilibrate. So the mutations that could change the duration of this time window would be the mutations which actually change how R star anagnin, the activated form, will influence the concentrations of NAHES and Vignin. So these mutations will change the equilibrium, which will change the time window. Also certain mutations which increase the rate of phosphorylation can also change the time window that is formed. Okay. So that is for the first two parts.
Three minutes remaining.
For the last part, as was visible from the enzymatic diagram, so the competent cells do not secrete pyridotin. So it will only be the sporulating cells that secrete pyridotin. And since pyridotin can cross membranes, so pyridotin secreted by one sporulating cell will go to its nearby cells as well. So the nearby cells will also receive pyridotin, and pyridotin would inhibit Srishti, which degrades S-anagnin. So what will pyridotin do is basically indirectly it will increase the production of S-anagnin, thereby incentivizing cells to become competent. So the evolutionary advantage it gives us, say suppose we have one cell that is doing say sporulation, then it will secrete pyridotin, which will encourage the other cells around it to do competence, which will so therefore you are actually increasing your own genetic diversity while ensuring reproduction as well. You're increasing the population and you're increasing your own genetic diversity, which is a pretty cool evolutionary advantage which is given to it.
30 seconds remaining. IT Kanpur, time is up.
So for the part B, where we have to explain the production of S and again. Basically, we have the sigmoid graph for the production of S and again in respect to concentration of S and again, and the linear shifted graph. So basically, if we subtract both of these for the making of S and again, if the line is after the sigmoid graph, it will have a positive area in the subtracted graph, which means S again and again is increasing, and hence will reach the competence stage, but it should be down. For the if the linear function is before the sigmoid, then it will go down and come up. So the area will be net 0, maybe 0, but it starts from negative. So S and again won't be being produced, and hence we won't reach the competition stage. This occurs because the initial concentration of Srishti may be different for different bacteriums, and that's the case.
So for the part D part, pyridotin supports competence. So if the sporulating cells secrete pyridotin, the surrounding cells will be can be competent. And the evolutionary advantage is that when you are competent, that time you will have a variety of gene pool, and you can survive. You might have a situation such that you have a gene so that you can survive in this stressed environment, and you can go into your, increase your progeny. Yeah.
IISER Bangalore, time is up.
Yes, so questions 1 and 4 we pretty much agree with the other teams. We'll expand on 2 and 3. In question 2, what sort of mutations can change? So if the inhibition of nihase or the promotion of vignin by R antigen is changed due to mutations in both of them, then that could change the time window. Or if the, and now for question 3.
For question 3, the S-anagnin is either both degraded and produced simultaneously. So we draw the graph for both of them, and where there's an intersection, the rate of degradation and the rate of productions are equal, and hence there is an equilibrium concentration. Now, if that equilibrium concentration of S is greater than that of R, then the competent cells dominate, and if it is less than R, then sporulation occurs, and hence we can't say that all the competent cells will be in the populations.
IIT Bombay, your time is up.
Yeah, so for part C, why doesn't every bacteria in the population become competent? We know that for competence requires that bacteria absorb the DNA that's released by other bacterium into the environment, and since there will not be much such in its environment, every bacterium cannot decide to become competent in this case. And yeah, as for the part A, the time window exists because R antigen can also activate vignin, or either it can, it also inhibits NEHASE, both of which inhibit this. Now, if the natural secretion of NEHASE or vignin can change depending on mutation, which would in turn change the amount of time that is allowed that for the bacterium to decide which way to go.
Till your time is up. Judges, do you have any questions for your teams?
We perceive any role for noise in the system? Never mind.
I would request the question makers to come and present the solution.
So for the noise part, we believe the equilibrium where both of them interact. So if there is a slight perturbation around that point, then equilibrium won't be stable, and we would either see it go all along to zero, or like all the cells would become competent.
Yes.
The noise part, we know that this NEHASE and vignin, so they together can inhibit this anagen. So if you have a noise, we know that our anagen inhibits NEHASE, so that is the one which can drive the competence. So if you have a noise and you destroy this, the competing effect of vignin and NEHASE, so that can induce competence in this.
Also for the B part, we can for the curves we made.
Your time is up. You can't
answer. Hello. Yeah, so overall, with all the 4 teams combined, you people nearly got the answer, so very well done. I'm very happy with you all. So this question is based off the real life phenomenon of Bacillus subtilis and sporulation and competence in the bacterium Bacillus subtilis. And all these proteins are real proteins with just the name swapped. First question as to how the time window forms. So the R anagen has different affinities for NEHASE and vignin. So if R anagen has a higher affinity for NEHASE as compared to vignin, so as the concentrations for R anagen rises, it will first inhibit NEHASE, and it will need to rise a bit further until its concentration is high enough to inhibit vignin. So there is a period of time when the concentration is between these two thresholds that NEHASE is inhibited but vignin is not activated. That's a time period during which, that's the only time period in which S anagen can rise in its rise beyond zero, that is. So this is how the time window is formed by different affinities or different thresholds of inhibition and activation. As for the mutations, this is fairly straightforward. Any mutation that affects the affinities, thresholds, any mutation that affects the rate of phosphorylation, anything like that, anything of that sort will change and affect the time window. As for the next part, this S anagen positive feedbacks upon itself to create a sigmoidal graph. and the negative feedback, if you overlap it with like this, overlap them this way, we end up with 3 equilibria points. Like when the production and degradation rates overlap, as correctly stated, they become equilibria points. So these 2 points are stable equilibria. Well, this point is an unstable equilibrium. So as S energon first rises, it will reach this stable equilibrium. And to become competent, it has to reach this, it has to reach this equilibrium. One thing that teams ought to have focused upon was that if this was a very well-tuned system, then every bacterium will get stuck at this equilibrium and will never become competent. So this is where noise comes in. So noise causes oscillations in the concentrations of all proteins, such as random oscillations. During the time window, if all the oscillations remain restricted to this region, like less than the unstable equilibrium, then the S energon concentration will always come back to the first stable point, and the bacterium will not become competent. It will sporulate. If before the time window closes, if random noise happens to cross the unstable equilibrium, then the positive feedback kicks in, and S energon will rise all the way to the second stable point, and the bacterium can now become competent. So it is in a sense a molecular coin toss, molecular equivalent of a coin toss. So either stable point and you get competence or sporulation. And finally, the paradictin part. So the answer we were intending, the answer we originally intended was that paradictin will act like a quorum sensor. So all cells are producing it at a constant rate, and it degrades very fast. So the concentration of paradictin will be directly proportional to the population density. So if the population density of the bacteria is high, paradictin is high, and Srishti is low. As a result, this graph, this line like shifts a bit over here. So the stable equilibria and the unstable equilibrium comes close. And this increases the probability of noise pushing them over. So higher population density, higher chances of competence, and vice versa. Less population density, graph shifts over here, the equilibria points shift apart. So the evolutionary significance is that if the population density is high, then only then it makes sense to be competent, because only then will the DNA in the environment be in a good enough concentration. If the population density is low, there's no point in becoming competent because there's less DNA to pick up anyways. However, the answers which we got, like a sporulating cell can force its neighbors to become competent, that's also like an evolutionary feasible answer, and we can accept that as the alternatively correct answer. Both make sense. As for finally, why do competent cells not release paradictin? It's simply that if all the cells in the population are to become competent, then there won't be any DNA to pick up. So you have to maintain a proper ratio of sporulating to competent cells. So as the cells in the population start becoming competent more and more, the concentration of paradictin falls, and the graph shifts upward, pushing the points apart. So basically, as cells become competent, they inhibit other cells from becoming competent. So that there's a good ratio of competent to sporulating cells. In reality, it's like 90% sporulating and 10% competent. So I hope you all learned something new.
So that would be the end of Biology Deep Thought Mimamsa Mains 2024. I thank you all for joining us today. And now I hand over the mic to our JC.
Yeah. So this ends the Biology Deep Thought. I want to thank every one of you for being present here. For the audience, I want to thank Professor Vinita and Dr. Suhita for being judge here. Now we are having snacks outside. Everyone please have these snacks, and we will be at 11:30 sharp. We will start Chemistry Deep Thought immediately after this. Teams, please go with the participants. Okay, thank you everyone.